Good morning, everyone. We, the Group 2, will going to discuss about fat storage and utilization and the biosynthesis of fatty acids or lipogenesis. And I'm Danica Fepernya. We'll tackle first about fat storage and utilization. As mentioned in our previous lesson, fat is our main source of energy, and our bodies need it to absorb fat-soluble vitamins and to protect organs. Fats or triacylglycerol are stored in adipose tissue, which is widely distributed in the body. Subcutaneous fats tends to gather under the skin or in the abdominal region and some of internal organs, especially the kidneys. So, fat deposit around internal organs which absorb a sudden impulse of energy, and these absorbers are called cushion. Subcutaneous fat acts as insulating blanket, which insulate the body from extreme temperature and helps keep the internal climate under control. Mammals that found in cold water like seals, walrus, and whale are examples of animals where a fatty acid developed. Fat is the major reserve of potential energy, and it metabolizes continuously. The stored fat does not remain in the body unchanged. So, there is an exchange between the fats of plasma lipoproteins and the fats in adipose tissue. The plasma lipoproteins are simple proteins and fats banded together to facilitate transport of the non-soluble fats through blood. And plasma lipoprotein that found in triacylglycerols or fats are broken down by the enzyme lipoprotein lipase that found on the walls of capillaries. Then the free fatty acids are transported into adipose cell. Then the fatty acids are converted back to triacylglycerol. When our body needs energy from fat, the adipose cell enzyme hydrolyzes triacylglycerols and fatty acids are exported to other body tissue, and these processes are under hormonal control. When there is more energy available in the diet than the body needs, the excess energy is used to make body fat and continue eating of more food than the body can result in obesity. That's all for my report. I hope you have learned something. And to discuss the biosynthesis of fatty acids or lipogenesis, here is Miss Andrea Faye Borromeo.
Good morning, everyone. I am Andre Fea Borromeo, and I will discuss about the biosynthesis of fatty acids or lipogenesis. What is lipogenesis? Lipogenesis is the conversion of fatty acids and glycerol into fats, or a metabolic process through which acetyl-CoA is converted to triglyceride for storage in fat. Simply, it is the biosynthesis of fatty acids from acetyl-CoA. It stores biochemical energy that is needed for metabolism. And it is the pathway by which all three of the major classes of nutrients are ultimately converted to body fat. The three major classes of nutrients are the carbohydrates, fats, and proteins, which is converted to body fats. What are the requirements of lipogenesis? Number 1 is the synthesis of fatty acids and glycerol. Number 2 is the activation of fatty acids by CoA and glycerol and the glycerokinase. Number 3 is the combination of activated fatty acids and glycerol to give triglyceride. Lipogenesis occurs mainly for the synthesis of palmitic acid. Palmitic acid is the most common saturated fatty acid found in animals, plants, and microorganisms. Lipogenesis occurs in the cytoplasm of liver, mammary glands, and adipose tissues. What is the process of lipogenesis? The process of lipogenesis begins with acetyl-CoA, which can be obtained from the catabolism of carbohydrates, fats, or proteins. Then, the next step is the subsequent addition of two carbon atoms from another acetyl-CoA. This process is repeated until fatty acids are the appropriate length. Lipogenesis occurs in the cytoplasm. The necessary acetyl-CoA is created in the mitochondria and cannot be transported across the mitochondrial membrane. To solve this problem, pyruvate is converted into both oxaloacetate and acetyl-CoA. Lipogenesis is the reverse of fatty acid oxidation or beta oxidation. However, fatty acid biosynthesis is not simply a reversal of fatty acid oxidation. That is all for my topic. I hope you learned something. To discuss the major difference between the fatty acid biosynthesis and fatty acid oxidation, here is Ms. Araojo.
Thank you, Ms. Borromeo. Good morning, everyone. I am Grace Araojo, and I will be discussing the major differences between the two pathways involving fatty acids, which are the anabolic and catabolic pathways. Just a quick recap from our previous discussion. Anabolism refers to the synthesis of simpler substances to more complex substances, while catabolism is the breaking down of complex substances to simpler ones. Going back, there are four major differences that were identified between these two pathways. First is that fatty acid oxidation, or catabolism, occurs in the mitochondria, while the fatty acid anabolism, or lipogenesis, occurs in the cytoplasm of the cell. Lipogenesis, or the anabolism, requires a set of enzymes that are different from the enzymes used in the catabolism of fats. The enzymes involved in the lipogenesis are acetyl-CoA, fatty acid synthase, and ATP citrate lyase, while in the catabolism, it involves the enzymes acyl-CoA dehydrogenase, anyl-CoA hydratase, hydroxyacyl-CoA dehydrogenase, and ketoacyl-CoA thiolase. Another difference is that in the anabolic pathway, the growing fatty acid chain bonds to a special acyl carrier protein, or ACPSH, which acts as a handle to transfer the grow long chain from one enzyme to another through the series of enzyme-catalyzed reactions in the pathway. Coenzyme A, or CoA, is the carrier in fatty acid catabolism. The fourth and last difference between the two pathways is that a preliminary set of reactions involving malonyl-CoA occurs for each two-carbon addition cycle in the synthesis. Malonyl refers to a three-carbon group and has no counterpart in the catabolic pathway. Malonyl-CoA is synthesized from acetyl-CoA and carbon dioxide in the presence of the enzyme acetyl-CoA carboxylase, ATP, and vitamin biotin. So in this chemical equation, again, the acetyl-CoA and carbon dioxide were able to synthesize malonyl-CoA with the help of ATP, biotin, and acetyl-CoA carboxylase. That is all for the major differences between the two pathways. I hope you were able to learn something about my discussion, and we will now proceed on the 5-step pathway or reaction.
These are the 5-step pathway or reaction. The first step is acetyl-CoA and malonyl-CoA bond to separate acyl carrier proteins. Malonyl-CoA is required for all the elongation steps of the fatty acid biosynthetic pathway and is formed by carboxylation of acyl-CoA by acetyl-CoA carboxylase. ACC is constructed from 4 separate proteins and requires biotin as a cofactor. Malonyl-CoA is made available to the enzymes of fatty acid biosynthesis by its conversion to malonyl ACP by malonyl transaxylase. Acetyl-CoA is converted into malonyl-CoA by the enzyme acetyl-CoA carboxylase. The malonyl group is transferred to an acyl carrier protein to yield malonyl ACP. Malonyl ACP undergoes a condensation reaction with acetyl-CoA to yield acyl ACP with the concomitant release of CO2, or marked as reaction. The second step is acetyl ACP and malonyl ACP condense with loss of carbon dioxide or decarboxylation. Decarboxylation is a chemical reaction that removes a carboxyl group and releases carbon dioxide. Acyl-malonyl acyl carrier protein or ACP condensing enzyme prepared from extracts of Escherichia coli catalyzes the condensation of acetyl ACP and malonyl ACP to form acetoacetyl ACP. Mechanistically, this is a two-step process in which the group is first transferred to the ACP or acyl carrier peptide, and then to the cysteines as each group of the condensing enzyme domain.
This is the continuation of the 5-step pathway. The 3 steps that follow are approximate reversal of 3-step in the fatty acid beta oxidation. Step 3. Reduction. Hydrogen of carbon 3 beta oxidation of fatty acid keto group. Reduction of alkenes. Addition to hydrogen to a carbon-carbon double bond is called hydrogenation. The overall effect of such as addition is the reductive removal of the double bond functional group. The simplest source of 2 hydrogen atoms is the molecular hydrogen, H2. Reduction of the beta-keto group to beta-hydroxyl group with the NADPH reaction 5th of the catalyzed beta-keto ACP reductase. Step 4. Dehydration. Formation of the double bond between carbon 2 and 3. Dehydration that happens between the alpha and the beta carbons. Reaction 6th in the beta oxidation of the fatty acid catalyzed by the beta-hydroxyacyl ACP dehydrase. And lastly, step 5, reduction. Hydrogen of carbon 2 and 3. Reduction of the trans double bond by NADPH, reaction 7th of the catalyzed by NLACP reductase.
Approximate reversal of 3-stage of fatty acid beta oxidation. The metabolism of fats has some features in common with that of carbohydrates. Acetyl-CoA produced in the catabolism of both carbohydrates and fatty acids can be used as a raw material for making other substances and as an energy source. Acetyl-CoA is oxidized via the citric acid cycle. More potential energy can be trapped in ATP. The ATP in turn serves as the source of energy needed for the production of other substances, including the synthesis of carbohydrates and fats.